So we are going to look at enzymes sharply. It's one of the topics that you should take note of during your this semester. All right. Although some questions on enzyme came out first semester, however, you're also going to see some few questions on enzymes this semester. So you should take important note of it and some of the things we are going to do here, while you also take your past questions, answer some questions on that same topic. All right. Now, we have the definition here. Of course, we know what enzymes are. Enzymes are biological catalysts. Of course, you know what a catalyst does. It speeds up the rate of a chemical reaction. I could still remember the definition of enzymes in modern biology, that enzymes are biological catalysts, proteinous in nature, that speeds up the rate of a chemical reaction without being used up during the course of the reaction. Right? So enzymes are biological catalysts composed mainly of protein. They are globular proteins. Enzymes are what kinds of protein? There are globular proteins. We have two types of proteins, globular and fibrous, based on this classification. But enzymes are globular proteins that speed up the rate of a chemical reaction without being used up during the course of the reaction. All right, but they remain unchanged at the end of the reaction and is free to interact again with more substrates. All right, now the chemicals or material which an enzyme work on is called the substrate. What the enzyme work on, the full substances, is called the substrate. If it works on lipid, in that case, lipid is the substrate. If it works on carbohydrate, carbohydrate is the substrate. Anything it works on, if it works on protein, protein is the substrate. Are you getting it? The full substance in which an enzyme work on, or the chemical material in which an enzyme work on, is called the substrate. Now, an enzyme combined with the substrate to form an enzyme substrate complex, which is short-lived. So you have the enzyme plus substrate, it forms an enzyme substrate complex. This enzyme substrate complex, two things can happen. It is either the enzyme substrate complex, if it has enough energy, that means if it has energy above the activation energy, the enzyme substrate complex will turn to enzyme product complex. Are you getting it? But if it does not have enough energy, this enzyme substrate complex is capable of going back to become an enzyme and a substrate separately. Are you getting it now? So if it has enough energy, the enzyme substrate complex will turn to enzyme product complex. Then the enzyme product complex will split to form an enzyme and the product. You discover that at the end of the reaction, the enzyme still remains. Are you getting it? The enzyme still remains. The initial enzyme that started the reaction still remains the initial enzyme, but however, the substrate has been converted to what? A product. That is why we said that enzymes, they remain unchanged at the end of the reaction. And one thing that you should particularly take note of is that you see that most of the reactions of enzymes are reversible reactions. It is in one of the past questions when they ask which of the following is not true about enzyme. All the enzymes reactions are reversible reactions. Is that okay? You must take important note of that, that all enzyme reactions are what kind of reaction? Reversible reactions. Is that okay? Now, the active site of an enzyme is the region that binds substrate molecule. That region that binds substrate molecule is the active site of an enzyme. Now, this is an active site. This is where the enzyme binds the substrate. What you have here is the enzyme. What you have here is the what? The substrate. This is also an active site. This is an enzyme. Now, this is the substrate for this active site. When they combine together, they form the enzyme substrate complex. You now discover that each enzyme is shaped in such a way that only its particular substrate can bind to it. For instance, the way this one is, this one cannot bind to this one here. Are you getting it? Why? Because the shape is different. The same way, this one cannot bind to this one. Why? Because the shape is different. Is that okay? So that is that for the enzyme substrate complex. Are you getting it now? And this property of the enzyme in such a way that it only out of specific substrate is called the specificity of an enzyme. Is that okay? That's what they call the specificity of an enzyme. Now, enzymes are classified according to their type of food they act on. We have amylases, proteases, and then we have lipases. Amylase, of course, it acts on amylose, breaking down amylose into glucose molecule. Proteases act on protein, breaking down protein into amino acid. Lipases act on lipid, breaking down lipid into glycerol and fatty acids. All right. These are the classification of enzyme based on the kind of food they act on. However, we are going to talk about a more generalized way of classifying enzyme as time progresses. Now, we said amylase act on carbohydrates, starch, and complex sugar, changing them to glucose, which the body can absorb. Some amylase enzymes are, we have maltase. Maltase break down what? Maltose into glucose and glucose. Sucrase break down sucrose into glucose and fructose. Lactose break down lactase rather break down lactose into glucose and galactose. And of course, Tyline is a salivary amylase that break down complex food in the mouth into simpler food like dextrin, dextran, and so on and so forth. All right. So that is that. Now, proteases act on protein, changing them into amino acid. Of course, you can see pepsin convert protein into peptone. Rennin coagulate meat protein. Trypsin convert protein to peptone. Erepsin convert peptone to what? Amino acid. All these on their proteases. I don't think there's any need for you to go deep into all this. All right. Just know that the proteases, they are the enzyme that convert what? Protein into the amino acid, and we have different category of them. We have peptin, rennin, trypsin, erepsin, and so on and so forth. Of course, we say lipases act on fat and oil, changing them into fatty acids and glycerol. I have explained that initially. Now, what are some of the properties of enzymes? We said all enzymes are globular proteins. They are not fibrous proteins, so you must take cognizance of that. All enzymes are globular proteins. Being proteins, they are coded by the DNA. You know, all proteins are coded by the what? The DNA via the central dogma of molecular biology, where DNA is converted to what? RNA and then to protein. Where we said that the process of converting DNA to RNA is transcription, then RNA to protein is what? Translation. Then RNA back to DNA is reverse transcription. All right, but being proteins are coded by what? DNA. They are catalysts which speed up. Now, they could also act as inhibitors. Are you getting it? They are catalysts which speed up, or inhibitors which slows down the rate of reaction within the body of the organism. But most times they usually act as catalysts, catalyzing various reactions. Their presence does not alter the nature and property of the end product of the reaction. They are very efficient. All these are properties of enzyme. Now, we say enzymes are also highly specific in action. I've explained that. That is the specificity. And enzymes act best within a narrow optional range of temperature and pH. Enzymes catalyze reactions are usually reversible reactions. I have stated something like that before. You can see starch is broken down to sugar, and the enzyme that does that is what does this. Is a reversible reaction. Emphasis on this, they are usually reversible reaction. In the exam, they might just ask you, which of the following is not a property of enzyme? And they say enzyme catalyzed reactions are irreversible reactions. You know that that one is a false. Enzyme catalyzed reactions are reversible reaction. Now, they remain chemically unchanged at the end of the reaction. Enzyme lowers the activation energy of the reaction they catalyze. We have explained that before, that they work by lowering the what? Activation energy. All right. Activation energy is more like the cutoff mark. If cutoff mark in NDU is like 250, some of you will still be at home. You wouldn't have reacted or wouldn't have been in the university. So what the enzyme do, the activation energy is like the energy that must be reached for a reaction to take place. It's like a cutoff, a threshold energy. Once a reaction reaches that energy level, it can take place. But if it does not reach that energy level, it will remain like that. So what the enzyme does is, let us assume they set that energy threshold as 250. What the enzyme does is to reduce it to like 100. Between 250 and 100, which one is easier to reach? Of course, you agree with me that 100 is easier. So for enzyme catalyzed reaction, immediately you reach 100, reaction will take place. Why for the one that is not enzyme catalyzed, if you reach 250 first? So this one will easily take place as compared to this one here, which is an uncatalyzed reaction. Now, we say enzyme possess active site where the reaction take place. This active site has specific shape, which makes the substrate enzyme specific to specific substrate, and that gives or ascribe enzymes with the what the status of specificity. They are specific to specific substrates. Enzymes are retarded by poison or inhibitors. There are certain things that can actually inhibit enzyme catalyzed reaction. These are either known as poison or we could call them what inhibitors. Now, some enzymes are inactive and therefore require coenzymes or agents to activate them. Example of such enzyme is the trypsinogen, which require enterokinase to be converted to what? Trypsin. Another example is pepsinogen, which require dilute HCl to be converted to what? Pepsin. Pepsinogen, when it is released, if e reach the stomach and the dilute HCl, the acidic nature of the stomach act on it, e become pepsinogen. Other examples are the renin angiotensin system, where you have angiotensinogen, where if it comes in contact, it becomes what? When it comes in contact with renin or something, it becomes angiotensin 1, which later to be converted to angiotensin 2. You guys are going to do that in your first semester physiology, something like that. So those enzymes, they actually need something. They are inactive. Whenever you see this word ogene attached to an enzyme, know that at first it is inactive. You need something to activate it. So you have something like trypsinogen to trypsin, pepsinogen to pepsin, angiotensinogen to what? Angiotensin. Is that okay? Exactly. Now, cofactors are coenzymes. There are very important things that actually they help in enzyme catalyzed reaction. A cofactor is a non-protein chemical compound or metallic ion that is required for an enzyme to function as a catalyst. The rate at which enzyme catalyzed reaction is called the enzyme kinetics. The rate at which the enzyme catalyzed reaction takes place is called the enzyme kinetics. All right? We said a cofactor is a non-protein chemical compound or metallic ion. Emphasis on non-protein. The cofactor is non-proteinous. You know enzymes are proteinous. However, the cofactors, they are non-proteinous chemical compounds or metallic ions that is required for an enzyme to function as a catalyst. Are you getting it? And the rate at which enzyme catalyzed reaction takes place is called the enzyme kinetics, more like the way we have chemical kinetics. Now, cofactors may be metals or small organic molecules, and their primary function is to what? Assist in enzyme activities. Take proper note of that. Their primary function is to assist in enzyme catalyzed activity. That is the main functions of the cofactor. Now, they are able to assist in performing certain necessary reactions that the enzyme cannot perform alone. Hence, they are like auxiliary. They offer help. They are auxiliary to the enzyme itself. Sometimes we call some of them coenzyme. They work with the enzyme. Example of such cofactors, we have vitamins, minerals, and ATP, adenosine triphosphate. Now, types of cofactor. Now, cofactors are divided into two. We have the organic cofactors, basically derived from vitamins, like we said earlier, and we have the inorganic cofactors or the ions. The organic cofactors are divided into two based on the kind of bond that is formed between the cofactor and the enzyme itself. Now, the organic cofactor, we have what we call coenzyme and prosthetic group. Emphasis on this. Organic cofactors are two. We have the coenzyme and then we have the prosthetic group. Then we have the inorganic cofactor. What distinguishes coenzyme from prosthetic group? Now, the coenzyme, they are loosely bound to the enzyme and may dissociate from the enzyme after reaction. These are the coenzyme. They are loosely bound. The differences between coenzyme and prosthetic group is that coenzyme, they are bound loosely to the enzyme, and after the reaction takes place, they will leave. While the prosthetic group, they are bound irreversibly. Like that means they are bound together with the enzyme for a very long time. Is that okay? That is the major differences. So the coenzymes, They are loosely bound to the enzyme and may dissociate from the enzyme after reaction, e.g. NAD+. So you should take note of these examples. We have NAD+, which participates in redox reaction, then coenzyme A site transfer reaction, then vitamin-derived coenzyme like TPP, derived from thiamine pyrophosphate, derived from vitamin B1. All these are examples of coenzyme. We also have FAD+, which also participates in redox reaction, flavin adenine dinucleotide. All those ones, they fall under the category of what? Coenzyme. Is that okay? Now, sorry, flavin is for what is it called? Prosthetic group. Now, for prosthetic group, they bond tightly or covalently to the enzyme, e.g. the hem found in hemoglobin and cytochrome that gives them their color is a prosthetic group, but there's also a cofactor. Flavin adenine dinucleotide, which is FAD, found in flavoproteins, are also prosthetic group. However, the question that may come is, which of the following is not a cofactor? You should know NAD+, coenzyme A, vitamin TPP, and FAD, HEM, all of them are cofactor. But you should be able to distinguish between the ones that are coenzyme. Coenzyme is NAD+, coenzyme A, then TPP, thiamine pyrophosphate, derived from vitamin B1. Then the prosthetic group, we have HEM, found in hemoglobin and cytochrome, of course, like the cytochrome P450, and flavin adenine dinucleotide found in flavoproteins. All right. Now let us look at some definition of certain terms. We have what we call isoenzyme, holoenzyme, and apoenzyme. What distinguishes these enzymes? Let us look at them. Now, isoenzyme, as name implies, isomer has to do with different forms of the same compound, right? So isoenzymes are different forms of the same enzyme that catalyze the same reaction, but they vary in their structure, kinetics, and tissue distribution. That is an isoenzyme. More like isomer. Compounds that have the same molecular formula but different structural arrangements. Same thing with isoenzyme. They are different forms of the same enzyme that catalyze the same reaction but vary in structure, kinetics, and tissue distribution. Then we have the holoenzymes. The holoenzymes are the active form of an enzyme, consisting of the apoenzyme, the protein part, and its necessary cofactor or coenzyme. Is that okay? That is the holoenzyme. Is that okay? Then we have what we call the apoenzyme. The apoenzyme is the inactive protein part of an enzyme that requires binding with cofactor or coenzyme to form the active holoenzyme. The holoenzyme now is like equals to apoenzyme plus cofactor. Is that okay? Apoenzyme plus cofactor, you form the holoenzyme. Is that understood? Why apoenzyme standing on its own is the inactive form of it. Is that okay? So that is the differences between them. Now, importance of enzymes. Of course, you know enzymes, they speed up chemical reactions in our body. They are vital for proper functioning of the liver and other organisms in the body. Too much or too little of certain enzyme can cause health problem. Enzymes in our blood can help health care provide check for injuries and disease. For instance, ALT, AST, ELISA for HIV, then ALT, alanine transferase, and the aspartate transferase. All these ones, they are used to test for what? For liver diseases. All right, liver diseases. These ones, they are liver enzymes. Once they are present inside of your blood, you know that you are having a liver issue. All right. So all these, they can actually help provide feedback for injuries and diseases. Do you get it? Exactly. Now, they are essential for respiration, digestion, muscle, and nerve functioning. The food you take, they are broken down by enzyme. Respiratory process for release of energy. Each of the steps are catalyzed by various enzymes. When you talk about glycolysis, Krebs cycle, breakdown of glucose to give us energy in form of ATP. All those processes are actually what carried out by enzymes, or they involve enzyme to catalyze. All right. Now, the classification of enzyme. The enzyme commission is a system of nomenclature that classify enzyme based on the reaction they undergo. An enzyme commission number consists of 4 parts separated by periods. Each provides specific activity about the enzyme function. Now, the enzyme commission, you have it as something like this: EC111. The first number talks about the class of the enzyme. The second number, the subclass. The third number, the subclass. And the fourth number, the serial identifier. But the most important thing you should know is the class of the enzyme. We have class 1 to class 6. You should be able to distinguish between which enzyme is in class 1 and which is in class 6. Although the notes I made here actually made a mistake under that one. All right. I actually made a mistake based on that one. Now let us look at, I said there are 6 primary types of enzymes. The 6 primary types of enzymes are, we have the first one. Class 1 is oxidoreductase. So when you see any enzyme starting with EC1, any enzyme that starts with EC1, know that that enzyme is oxidoreductase. That means it is involved in what? Oxidation reduction reaction. Oxidation and reduction reaction. Then class 2, EC2, is transferase. They transfer functional groups such as methyltransferase and phosphotransferase. So any enzyme you see that starts with EC2, know that it's a transferase enzyme. Then 3 is hydrolase, EC3. They catalyze hydrolysis, that means breakdown with water. Any enzyme that you see start with EC3. For instance, you may have an enzyme like this, EC31111. Know that this particular enzyme actually break down something. You see them like esterase, cholinesterase. This one, they break down something. Then the fourth one is the lyases. This one, they add or remove groups to form double bond, like the pyruvate decarboxylate enzyme. So if you see EC4, know that that enzyme is a lyase enzyme. All right. Then number 5, this is where the mistake is. All right. Number 5 is supposed to be isomerase. So please take note of this. Isomerase, which is EC5. And this one catalyze isomerization. You know what isomerization is all about now. Then the last one is ligases, which is EC6. So EC6 is ligases. And that is one of the things I should. These ligases, they join 2 things together. All right. They join 2 things together. However, there is a 7th one, which is called translocase. That one is just added recently. There's a 7th one, which we call translocase. It involves the movement of ions or molecule across membrane. All right. So please take note of this. These things are very, very important. So you may be given any question. Enzyme that catalyze the isomerization or isomerism is called what? Isomerase. You know what hydrolase? Breakdown using water, hydrolase. Oxidation reduction reaction, oxidoreductase. And you should know their enzyme classification number. Know number 1 to 7, which of them fall in as 1 and something like that. All right. So I usually use OTH, LIL. OTH, LIL. Oxidoreductase, transferase, hydrolase, lyase, isomerase, ligase. All right. Take note of that. Now, enzyme inhibition. We could have competitive inhibition and we could also have non-competitive inhibition. Now, we'll talk about competitive inhibition. What we mean is that the inhibitor bind to the active site of the enzyme. You know, we said every enzyme has an active site that is shaped the same way as the substrate. So what happens is that the competitive inhibitor, instead of the competitive inhibitor to just stay, it will go and bind to the substrate. It will go and bind to the active site of that enzyme. Now, if it binds to the active site of that enzyme, what happens? It will prevent the substrate from binding, thereby inhibiting the enzyme substrate reaction. That one is a competitive inhibitor. While the non-competitive inhibitor is the one that will not bind to the active site, but it will bind to any other site. For instance, it bind to the allosteric site of that enzyme. And once it binds to that allosteric site, that means any other site apart from the active site of the enzyme, it will now change the shape of the active site of the enzyme, thereby preventing the substrate from binding. Are you getting it? One, competitive bind to the substrate direct. The non-competitive binds to somewhere else, but its binding to that place now will now change the shape of the active site of the enzyme, and the substrate can no longer what? Attach. Are you getting it now? So that is the difference between competitive and non-competitive inhibitor. Now, we have the following question. Which of the following is the main function of enzymes? The main function of enzymes is to catalyze biochemical reaction. Enzymes are primarily composed of protein. Then the part of the enzyme where the substrate bind is called the what? Active site. That's the part of the enzyme where the substrate bind. Which of the following factors does not affect enzyme activity? We have temperature, pH, substrate concentration. The color of the enzyme does not affect enzyme activity. Then, what is a cofactor? Cofactor is a non-protein chemical that assists enzyme function. We said that before. Which of the following is an example of a coenzyme? Take note, a coenzyme, we have NAD+. All these magnesium ion, zinc ion, chloride ion, they are what? Under inorganic cofactors. Now, the enzyme that breaks down hydrogen peroxide into water and oxygen is known as catalase. It's known as what? Catalase enzyme. Enzyme specificity refers to the enzyme's ability to be inhibited. No, the enzyme's ability to bind to a specific substrate. Should be. Now, what type of enzyme catalyzes the transfer of phosphate group? I said anything that catalyzes transfer is called a transferase of any group. It can be methyltransferase, it can be phosphate transferase. Is what? A transferase enzyme. Which enzyme classification involves the breaking of bond by addition of water? Hydrolase. By addition of water is hydrolase. Then, which enzyme is responsible for the digestion of protein in the stomach? That one is what? Pepsin. Right? And then, which enzyme break down starch into simpler sugar? Of course, you know, is amylase. All right? Then, enzymes that catalyze the same reaction but have different structures are called what? They are called isoenzymes. Then the next one, which enzyme help in digestion of fats? Fat has to be what? Lipase. And then the last, second to last, what does the term enzyme denaturation mean? Now, when we talk about enzyme denaturation, we are talking about the loss of the enzyme structure and function. Is that okay? Enzyme denaturation has to do with the what? The loss of the enzyme structure and function. That is what denaturation is all about. All right, which is option C. And then enzyme that catalyze the reaction, but we've answered this one before, number 40. And that is everything for this particular one. Thank you very much. We'll see in the next one. Catalyze the same reaction but have different structure are isoenzymes. That makes option A the correct answer. Now, we've talked about competitive and non-competitive. Now, example of some competitive inhibitors, we have methotrexate. These ones, they are drugs. All right, they are drugs for cancer, cancer therapy. Then statins, they are lipid drugs. All right, to reduce lipid lowering drugs. Then sulfonamines, antibiotics. Viagra, sildenafil is an enzyme inhibitor. Then we have Viagra, sildenafil. This one is for erectile dysfunction. Captopril, ACE inhibitor. This one, angiotensin converting enzyme inhibitor. Then we have ethanol and allopurinol. Allopurinol is used for treatment of gout. All right, and so on and so forth. All these ones, there are various drugs that can be used as enzyme inhibitors. Then we have this one, they are what? Competitive. That means they bind to the active site of the enzyme. Then we have non-competitive inhibitors. This one don't bind to the active site, but they bind to the, what is it called? They binds to the allosteric site or any other site apart from the active site, and thereby changing the shape of the enzyme. Example is alanine. Of course, you know alanine is an amino acid. Heavy metals like lead and mercury, they are non-competitive inhibitor. Doxycycline is an antibiotic. And then acetazolamide also. These are drugs. I think this carbonic acid and anhydrase inhibitor, carbonic acid anhydrase inhibitor. They are used for treatment of glaucoma. All right, glaucoma they reduce intraocular pressure. Is used to make most of some of these eye drops that are used for treatment of the silent thief of eye, which is glaucoma. All right, all those on they are non-competitive inhibitors. That means they don't bind directly to the active site of the enzyme. Now, other properties of enzymes are they are stereospecific, they are regioselective, and they are chemoselective. Stereospecific. Now, let's take them one by one. Enzymes are highly specific for the stereochemistry of their substrate and product. They can distinguish between different stereoisomers of a compound, often catalyzing reaction with one specific isomer under another. If you have two compounds that are different isomers, the enzyme can actually react with that specific isomer that it wants to react with. Let us say you have arrow and X. You guys have learned about E and Z, arrow and S in chemistry. Now, these are two the same compound, but they are different isomer. The enzyme is capable of knowing if it wants to act on the E, it can act on the E. If you want to act on the arrow, so it is stereospecific. Is that okay? Then the next one is regioselective. Now, enzymes can distinguish between different regions of a molecule and selectively catalyze reaction at specific sites. This means that they can favor the formation or modification of particular bond within a substrate. Regioselective has to do with position of a molecule and selectively catalyze reaction at specific site. They say they are regioselective. They're chemoselective. Now, enzymes can selectively catalyze a specific chemical reaction among many possible reactions in a substrate. Different reactions can be taking place, but enzyme may only catalyze just one reaction. Are you getting it? They can preferably act on one type of functional group in the presence of other functional group. You say they are what? Chemoselective. Now, we said enzyme catalyzed reactions are always reversible, irreversible, associated with energy loss, associated with energy gain. The correct answer is enzyme catalyzed reactions are always reversible. All right? It could be associated with energy loss, sometimes energy gain, but they are always reversible. Now, which of the molecule is a good example of enzyme cofactor? Example of enzyme cofactor. We have NAD plus is an example. Forget about this vitamin B1. It is the thiamine pyrophosphate from here that is the cofactor, but this one, NAD plus, is a good example of enzyme cofactor. Which of the following can inhibit enzyme reaction in organism? Now, herbicide, pesticide, they are enzymes inhibitor. This one can inhibit enzyme reaction in organism. All right. Sulfonamide and malolate are examples of, we said sulfonamide and malolate, they are examples of what? Competitive inhibitors of enzymes. All right. They are examples of what? And competitive inhibitors of enzymes. You can see it here, sulfonamide. They are examples of competitive inhibitors of what? Enzymes. So take note of that, which is option A, competitive inhibitor. The activity of enzymes are affected by pH and temperature, activation energy, active site cofactor. They are affected by pH and temperature predominantly. These are external factors. The cofactor, activation energy, and the rest, they are inherent within the enzyme, but pH and temperature are external factors that affect the activity of enzymes. Enzymes act by converting starting material known as dash into different molecules, known as substrate, into different molecules. Is that okay? Then the next one, like all catalysts, enzyme increase the rate of reaction by lowering its activation energy. I've explained that before. All right, when I say lower the cutoff from 250 to 100, and everybody rush into the university. Is that okay? Then the next one. Enzymes show impressive level of stereospecificity, radioselectivity, chemoselectivity. All of the above is the correct answer. Because I have also corrected this. Ligases are classified as ECCs because I told you that ligases are enzymes that join two molecules together, all right, with a covalent bond. I think enzymes convert reactants into product. Enzyme catalyze convert reactant into product, and I think that is all for enzymes. Let me see if I can still see some questions under enzyme from past question. Let's just look at all of them together at once. Here's a past question. Who sent me that past question the other day? Okay. All right. Most enzymes are protein. We said that. Okay, I think we have done justice to most of the questions here. Let me search for others. Okay, we have another one here that says, how many enzymes are known to us? I mean, to use the coenzyme NADH. How many enzymes are known to use the coenzyme NADH? What is the correct answer? How many enzymes are known to use the coenzyme NADH? I don't think there's a specific number to that, but I know that there are just over 500 enzymes, but specific number to that, I don't think. It's estimated that over 500. You have 900, 700, 600, 500. Over 500 should be around 600 also. So that is that. Now, which other question do we have here that has to do with enzyme? I think that is all. Okay, no, there are so many other questions. So coming. There's some total of all chemical reaction going on in a cell. They are called metabolism. Which of the following statement is not true of enzyme? Enzymes are globular protein. I've said that. They are catalysts, yes. Enzymes are specific in reaction, yes. All proteins are enzymes. That is lie. All enzymes are protein, but not all proteins are enzymes. So option D is not correct. Which of these factors does not affect the rate of enzyme reaction? A. Enzyme concentration. B. pH. C. Substrate. D. H plus. H plus is still more like acidity, but pH is already there, so H plus is not correct. All right, H plus is not correct. So that is that for that particular one. Let me look for more question. Attenuated organisms are used for This one is just a random question. Attenuated organisms are used for antibiotics, cream, vaccine, cough syrup. They are used for vaccines. All right. They are used for vaccines. All right. Then antibody play an important role in cellular immunity, innate immunity, humoral immunity, and then lymphoid cell. They play an important role in humoral immunity. That's where you have immunoglobulin. All right. So you should be able to know all those ones. Those ones they are under immunity, which we looked at. And I believe that is all for now.